individuals have T cells at various differentiation stages. We do have naive T cells, effector memory T cells, and we also have a small population of double negative T cells, DNT, which lack CD4 and CD8 expression. And all these distinct subsets have different phenotypes, and they can expand or contract, for example, during infection, but overall they are kept under tight homeostasis. In a pathological condition, however, which is called ALPS, which stands for autoimmune lymphoproliferative syndrome, the maintenance of this homeostasis is defective, and DNT with a very unusual phenotype, you can see here in red, massively expand and lead to a huge lymphoproliferation in the patients. We aim in the lab to understand what is molecular basis of this uncontrolled T cell behavior. And ALPS as a disease is most frequently caused by mutations in the FAS gene, and FAS is highly expressed on all activated T cells. So at the beginning, for this reason, it seemed likely that DNT in ALPS simply derive from activated T cells. However, it was actually already known by previous studies, mainly based on flow cytometry, that ALPS DNT actually express, for example, also markers of naive T cells, making this hypothesis not conclusive. And we wanted to clarify the subject, and the first thing we did is to further characterize the population. So we sorted DNT from 3 ALPS patients, and we sorted also CD8 cells. On one side, naive and early differentiated here, CD28 positive and 57 negative, and late differentiated CD8 cells, CD57 positive and CD28 negative. And we sorted also late differentiated CD8 cells also of 3 healthy individuals. And we performed RNA sequencing analysis. And as you can see here, the transcriptional profile of ALPS DNT was unique and different from the other analyzed T cell subsets. And in details here, you can see the number of differentially expressed genes in every comparison we made. Having a closer look at the differentially expressed genes, we did find molecules already known to be dysregulated in ALPS DNT, for example, IL-10 here and FAS ligand upregulated, but also CLARG1 and TBET strongly downregulated. Interestingly, we found a lot of genes encoding for galactins and glycosylation enzymes. And this is interesting because it was already known that ALPS DNT express high levels of B220, which is a glycoform of CD45R, result of a high glycosylation activity. We found also a potpourri, I would say, of markers typically expressed by activated cells, I say here CD38, but also by exhausted cells or regulatory T cells, without, however, finding key similarities leading to a strong link with a known T cell subset. And just to give you a number, what we found also are 746 genes that we called DNT private genes, because they were dysregulated not only in the comparison with the naive and early differentiated, but also with the late differentiated T cells. And among these private genes, we found also some transcription factors, and I point out here for you MIB, strongly upregulating, and OBF1 here. We then moved to the protein expression profile, and we performed mass cytometry analysis with samples of 7 ALPS patients and 7 healthy donors. And we saw actually that there were 2 phenotypically distinct DNT populations. One of them expanded in the ALPS patients here in red. And these cells were actually highly expressing CD38, but also CD45RA, CD27 and N28, but also CD57, Some of them were highly expressing Ki67 and HLA-DR, but also CTLA-4. And we termed these cells particularly expanded in FAST-deficient patients, FAST-controlled DNT, so FC-DNT. And you can see the population probably even better here. You see a t-SNP plot, this time showing only DNT, TCR gamma delta negative DNT. And you see here in red the fast-controlled DNT, and in blue this other smaller DNT population clearly present also in the healthy donors, which we termed conventional DNT. So we performed on all the DNTs used in the site of experiments, both of the patients and the DNT and the healthy donors, and we performed unsupervised clustering approach termed flowsome, and we were happy to see that this also generated two main DNT clusters, cluster 1 and 2, which are the FC DNT, and clusters 3, 4, and 5, which are the conventional DNT. And to visualize the relationships between these clusters, we used another approach, which is called UMAP. And this was also generating two main population, here 1 and 2. And it was also nice to see that when we projected the 5 clusters found by flowsome to the UMAP plot, they were matching very nicely. And then we knew already from the site of data, here once again, that the FC DNT were most homogeneously expressing CD38 and CD45RA. So we wanted to see if the combination of these two markers was able to divide efficiently FC DNT from cDNT. And so we projected the two populations found by manually gating with these two markers on the UMAP, and it was nice to see that it was projecting correctly to the two different subpopulations. And this was really useful because this meant that we could reduce a multi-parametric definition found using all the markers we had used in the site of experiment to a two markers definition of these two populations, which could make possible the finding of these cells also using simple flow cytometry analysis. And then last piece of information of this figure here, figure 2H, you can see when you project the two DNT population to a UMAP showing all the T cells, so CD4, CD8, and the TCR gamma delta T cells, you can see that the FC DNT make up a distinct population, whereas the cDNT align more together with CD8 and gamma delta. Again, based on this very specific experiment made with the markers you are seeing here. We were, however, actually here, I go back to this part of figure 2 because these very few red dots here, you can find here, we were actually not expecting that. This was the serendipity moment in the project. So we were actually thinking disease versus healthy. So we were definitely not expecting to find some FC DNT also among the healthy donors. But it was, of course, a very interesting finding, and we wanted to follow up on that. So we performed very many flow cytometry analysis on many healthy individuals, and we could indeed find out that the FC DNT are present not only in ALPS patients, but also in healthy individuals. And in these healthy individuals, they share important features with the FC DNT of the ALPS patients. For example, they lack expression for TBET and for CLARG1, but they have high expression of Ki67, EOMES, B220, CD57, and as already noted, CD27 and 28. We confirmed the similarities also on a transcriptional level. We performed RT-PCR of some of the key signature genes. You can see here IL-10, OBF1, MIB, and galectin-3. And we could see that not only the Fc-DNT of the patients, but also the Fc-DNT of the healthy donors showed upregulation for these genes. And this was true also on a protein expression level. You can see here IL-10, phospho-MIB, and galectin-3. So this was a very interesting finding, and we were surprised. And so we wanted to see closer. So we wanted to explore further the relationship between the Fc-DNT in the healthy donors and in the patients, and their link with the conventional DNT. So we went back practically to transcriptional analysis. We sorted again, and we sorted of healthy donors, naive and early differentiated CD8 T cells to use as a reference population. So exactly the same population sorted also in the first transcriptional analysis you saw in figure 1. And then we sorted Fc-DNT and conventional DNT based on the expression of CD38 and CD35Ra. And as you can see here, there was more or less 1/3 of the genes which were similarly upregulated in patients and healthy donors. 1/3 of the genes were similarly downregulated, and these genes up and down included some of the key signature genes. But there were also some genes actually not concordant in the dysregulation. And we wanted to know which are these genes. So we performed gene set enrichment analysis here, and we saw that the pathways involved in this difference were mainly pathways of cell cycle progression, glycolysis, and mTOR signaling, possibly suggesting that this difference was an activation state difference more than something else. And we wanted also to confirm it on a protein expression level. So we chose 2 markers, HIF1 alpha and GLUT1, which are known to be part not only of glycolysis, but also of mTOR signaling. And indeed, we could see that these were much higher upregulated in ALPS patients, Fc-DNT, than in healthy individuals. As I told you at the beginning, we were then again interested in better characterizing these cells. And we noticed at the beginning that Fc-DNT do express many markers of activated cells. I cited already CD38, HLA-DR, Ki67. So we thought about comparing these cells with in vivo, with other in vivo activated T cells. And so we collected blood samples of healthy individuals, so immunologically healthy individuals with acute EBV infection, and we concentrated on their CD8 T cells. And they were, as expected, as you can see here, very much expanded in green. And these CD8, they were, yes, expressing CD38 and HLA-DR, exactly like the Fc-DNT. However, they were also highly expressing CLARG1 and TBET, which was not the case in Fc-DNT, and they were definitely not expressing CD45RA and B220, in contrast to the Fc-DNT. We checked also once again with RT-PCR the key signature genes. Now you know them probably, IL-10, OBF1, MIB, and galectin-3. And everything which is expressed by EBV CD8 cells should be here green, and you don't see actually anything green, which means these genes were really not upregulated in these cells. And having made this comparison with the activated T cells led us to ask which is the cytokine profile of these cells. What can they do actually? So we checked cytokine production in different T cell subsets in vitro after stimulation with PMA-IONO. And we could see that the Fc-DNT were actually able to make IL-10, but they were not able to make anything else. And this included also the lack of expression of cytolytic molecules. We checked for granzyme B and perforin expression by flow cytometry, and we couldn't find anything. And interesting also in this slide is that on the other hand, the conventional DNT, they were able to produce interferon gamma after stimulation, and they were also expressing a bit of cytolytic molecules, making again them more similar to the CD8 actually rather than to the FcDNT. Then we moved to another important question we had since a long time. So we asked, is this signature actually defined by the DNT state, or is it present also in CD4 and CD8 T cells? And we knew already from previous studies on ALPS patients that ALPS patients do have a small proportion of CD4 and CD8 single positive T cells, which share some phenotypic features and also some transcriptional features with the DNT. And so we performed again transcriptional analysis, and it was very nice to see, so RNA sequencing, and it was very nice to see that the profile of the ALPS DNT was pretty much identical with the one of the DNT-like, in this case, single positive CD8 cells, which was true and nice also in the PCA analysis. We confirmed these also using cytoph and flow cytometry. So what you can see here is a t-SNE plot made without CD4 and CD8, only with the CD3 positive cells. And you can check among the cells expressing high levels of CD38 if among these cells there are only DNT or also CD4 and CD8 single positive T cells, which was actually the case not only in the patients but also in the healthy donors. And this was true also using flow cytometry. As you can see here, very, very, very small percentages in the healthy donors, single positive T cells, but very clear and expanded in ALPS patients, where you can see here the signature single positive T cells expanded and also actually very, very nice to gauge. Then a couple of information regarding this FcDNT. So we tried to understand them better. We looked for them, and we looked for them already in cord blood samples to see if they are present before birth. And the answer was yes. We found them in cord blood samples, and we found the signature resembling the one of the FcDNT in adult peripheral blood. We found them to be between 2 and 29% of the total TCR alpha beta positive DNT in peripheral blood, and we found that they are tendentially not increasing with age. Here we found them enriched in secondary lymphoid organs when compared to bone marrow and peripheral blood, and we found out that these cells are not expanded in the context of acute viral infection. Again, we checked in these samples of individuals with acute EBV infection here in green, and we found that these cells were not expanded in the context of a viral infection. Then one of the main questions we had since the beginning was, what is the role of FAS in all this story? We termed them FAS-controlled T cells, but what does it do? And so the first thing we checked was the expression level of FAS in FcDNT of healthy donors. And as you can see here, FcDNT do have FAS expression levels comparable to memory CD8 or CD4 T cells. So a difference in expression was not to see. So what we did, however, is we checked for FAS ligand-induced apoptosis, and we found that FcDNT are much more sensitive to FAS ligand-induced apoptosis, and this you can see here. So we measured after in vitro stimulation or stimulation, so incubation with the increasing concentration of Fc FAS ligand. We checked for percentage of active caspase 3 as an indicator for FAS-induced apoptosis, and you can see that the FcDNT, always in red, they are much more sensitive than the other analyzed T cell subsets. Another thing we knew already from previous studies is that ALPS DNT, so DNT in ALPS patients, they are mTOR dependent. And this is something we know also very well from the clinic, because ALPS patients are mostly treated with rapamycin for their lymphoproliferation. But the question we had was if this mTOR dependence was actually specific for FC-DNT. So we checked for phospho-6 ex vivo, and we found it upregulated in FC-DNT compared to C-DNT, both at baseline and after stimulation. And this was true also for the single positive counterpart, not only for the DNT. We checked then with Cytov the DNT of 2 patients with ALPS before and after treatment with rapamycin, and we could see a selective decrease in FC-DNT and single positive FC T cells. And that was also very cool. We could collect some samples of immunologically healthy individuals with no ALPS, but that were treated with rapamycin for other reasons, mostly actually vascular malformations. And we could see also in these sort of immunologically healthy individuals that giving rapamycin was leading also to a selective reduction in FC-DNT, as you can see here. So interestingly, one last important information of this slide is there was a selective reduction in the signature T cells, but there was not a change in the signature, which led us to think that FAS and mTOR were able to, the one control the population, the other promote the survival of the population, but not to drive their differentiation. And then the last, and this is really the last figure of the paper, we checked for FC-DNT also in other patients with other primary immunodeficiencies with lymphoproliferation, where it was already described that there was an increase in total DNT. And these diseases are STAT3 gain of function and CTLA4 deficiency. And we found also here moderate increase in FC T cells, both in STAT3 gain of function and in CTLA4 deficiency. And this was true not only for the double negative T cells, but also for the single positive signature cells here, CD4 and CD8 analyzed by flow cytometry. And for CTLA4, we knew already from older papers published on LPR mice, which are the ALPS mice, so to say, that when treating these mice with CTLA4 Ig, it was possible to reduce the lymphoproliferation, but B220 positive DNT were still found in the tissues and peripheral blood of the mice. So we knew that CTLA4 could, together with FAS, play a role in the control of this population. So we decided to concentrate a little bit more on the possible role of STAT3 signaling. So we checked ex vivo samples of ALPS patients or healthy donors, and we saw an increased phosphostat-3 in FC-DNT compared to conventional DNT, and this is again true also for the single positive signature T cells. We found a full responsiveness of the FC-DNT in vitro after stimulation with IL-21. And we come here to the probably the frustrating part of working with the DNT in the lab. So it is very well known that this DNT die very quickly ex vivo, which makes functional assays very hard. And so what we do in the lab is always try to find something which make them, keep them alive. And here we tried to add to a survival assay performed for 24 hours, also IL-10 and IL-21, so STAT3 activating cytokines to the medium. And we checked for the survival by staining for annexin 5 after 24 hours of incubation. And as you can see here is the starting time point, and here with medium alone, and here with IL-10 or IL-21. I mean, they still die, unfortunately, but you can see a sort of improvement in the survival when adding this STAT3 activating cytokine instead of by putting medium alone, or we tried also with IL-2 and it didn't help. So to sum up, we identified a unique signature which was present in DNT, but also shared by CD4 and CD8.